Okay.
Okay, I think you can start now.
Okay, great. I think I'm gonna share my screen right now because I have a few slides. Share screen. Here we go. Okay, so Kathy asked me to actually to give a career talk here. So I'm gonna speak a little bit about myself. So I'm Dr. Lin Jiang. I'm professor in the School of Biological Sciences at the Georgia Tech. So it's the full name Georgia Institute of Technology, and often called Georgia Tech. And for those of you who don't know Georgia Tech, it's located in Atlanta, Georgia. And so I'm, like I said, I'm in the School of Biological Sciences, and we have a relatively large group, I think between 70 and 80 faculty. And I'm one of the ecologists, and I'm a community ecologist. If you don't know what ecology is, it's very simple. I think everyone knows a little bit about ecology, and so basically it's studying the relationship between organisms and their environment. And I'm a community ecologist focusing on interactions between different species. So a little bit more about myself. So I was born and also grew up in China. So I went to Nankai University in Tianjin, China, for college. And then I went to Peking University for my master's. Then I moved to the United States and Rutgers University in New Brunswick, New Jersey, for my Ph.D. So it's always my major was always ecology related, and because from the very beginning I was very interested in ecology, particularly conservation biology. I thought I would, once I finish my PhD, I would probably work as a conservation biologist, but that's not the case because I don't work with the conservation on conservation biology. I'm a community ecologist and looking at the interaction between species, often working with microbes and plants. So I wanted to, when I was little, I wanted to work on with mammals, like endangered mammal species, but that's not the case. But still, I'm an ecologist. So that's my degree. So here, and I joined after 2 years after, actually 3 years after I got my PhD. I joined Georgia Tech as assistant professor. Then a few years in 2006. Then 2012, I was promoted to associate professor. And since 2017, I'm a full professor in the School of Biological Sciences at Georgia Tech. So this is just a picture of my lab webpage, and actually there's I think there's address is down here. So if you're interested in what I do, and then you can go to this website. And so this is my Google Scholar web page. So for those of you who don't know Google Scholar, basically it's designed for academic people, including like the faculties and students, postdocs, and here, and they listed all my publications. I think if not all, most of my publications here, and also like citations, how many times have been cited. H index, my current H index is 43. That means like 43 of my publications have been cited at least 43 times. And my I-10 index is 84. That means like 84 of my papers have been cited at least 10 times. So here, like last year was cited over 1,200 times. So this year, so up to now, it's about slightly less than 1,000 times. So then I want to walk you through some of my study systems as an ecologist, and we want to understand nature and to understand how nature operates. So that's what I do. Part of what I do, I work in the lab, both in the lab as well as in the field. So here is one of them, my field site, Southern Island Lake in China. And it does have over 1,000 islands. In fact, it has over 2,000 islands in this particular lake, and it's actually a reservoir. And I think the reservoir was formed back in the 1950s. So as a result, over 2,000 islands formed, and those islands differ in shape, in size. Some as small as just less than 1 acre. The biggest island is about, I think, over 5,000 acres. So there's a large gradient of island size. And then what we did was we were funded by National Science Foundation biodiversity dimension of biodiversity program, and to study biodiversity on those islands, not in the lake. So we work with the terrestrial ecosystem here, not the lake ecosystem. And so here is, so we want to understand what regulated biodiversity on those islands, including plant diversity. So here is the picture of my students. On the right hand side is my student, my former student, Lin Jiang. And on the left hand side is just the local fisherman, actually. And he, we rented his boat, so basically to visit one island, from one island to another. So they are basically taking leaf samples. And so we also studied leaf litter decomposition. So those bags are leaf litter bags. So we deploy them and then collect them back, retrieve them, and every month, and then monitor the decomposition rate of the leaf litter. And what we found out was actually the leaf litter decomposed faster on larger islands. So we also look at soil microbes. So here is a picture of my former postdoc. Here is Xiaopeng Li, who is now a professor at Eastern Normal University. And here, this is another fisherman. So they're collecting soil samples. So we collect soil samples, extract DNA from the soil. So looking at bacteria and fungal diversity. And what we found out was actually also the soil bacteria, fungal diversity tend to be greater on larger islands. And we also, of course, we studied trees. So we have plots, 20 meter by 20 meter plots on each island. So we tagged each tree, and then also we use the growth rate. We call it. It's a formally called a dendrometer, used to measure the diameter of the trees. And so it can use the diameter to estimate their growth. And finally, I want to show you the really cool caterpillar and we found on the Southern Island Lake Islands. And it's a specialist, actually, caterpillar feeding on the Chinese red pine. So it's the Chinese red pine is very, very common on those islands. So this it's one of the dominant early successional species on those islands. and actually it's an early successful species in this region. It's from subtropic China, and although we didn't study this caterpillar, and it was one of the pest species feeding on one of the dominant tree species on the island. This is the island study. We also study grassland. So this picture of the Inner Mongolia grassland. So I think it's a picture taken back in August. So you can see the biomass is really high. So biomass is actually the August is the grassland biomass is the highest in August, I think. And so we did a number in collaboration with the colleagues in China. So we conduct a number of global change experiment. For example, this is the warming experiment. So this is you can see here, the bar here is the infrared heater. So you use it to heat up the air. So we look at how warming affects the community structure of those grasslands, the ecosystem function of those grasslands. So we also look at other stuff. We also set up other experiments. For example, I think this is the dust experiment, and so we manipulate dust amount among different plots. So we can see. I think it's a 10 by 10 plot. So this is the Inner Mongolia grassland. We also work at in Tibet. So here is the picture of the Tibetan Alpine grassland. So in the back, you can see the Qilian mountain there. And then I took this picture back in May 2019. And here's another picture. So you can see Qilian mountain in the back as well, but it's, here a bunch of Tibetan domestic yaks, and those are essential for actually those are really important livestock for local Tibetan people. And we did experiment there studying the effect of adding nitrogen and also adding phosphorus into the soil, and also we also manipulate light availability. So what you see here is it's a picture taken from a drone. It's a drone picture of our experimental plots. So you can see the bar here are essentially LED lights. So this is the closer look of experimental plot, and then this is the close-up of experimental plot. So this is the second year of our experiment. We started last May, I think, and so we're hoping to continue experiment at least until next summer. So this is the field study. So we also do work in the lab. So this picture of our experimental microcosms. So basically really small ecosystems, and then you construct in the lab. So we call them laboratory microcosms, and we work with protozoa. So you can see here the different type of microcosms, and depending on what kind of a container you use, you have jars, you have test tubes, and you have also petri dishes. And so here so this type of work, the microcosm-based work, and came from a Russian ecologist, Gauss, and he in this book, The Struggle for Existence, is a classic book. In this book, he actually talked about a few experiments, and he did in the lab and working with protozoa. So just if you don't know what protozoa is, they are just single cell animals, okay? So like this here, Paramecium, you guys probably don't know Paramecium. It's a ciliated protozoa and but it's only single cell. No, they got no brains. So here is paramecium, and here didymium is also single cell, and so here is the paramecium is actually the prey for didymium. And if you click this YouTube link, we may be able to see the video of the prey, the predation going on here. Okay, so here's the video. You can see this is didymium, this is the paramecium, and didymium is actually a little bit smaller than paramecium. So here, the predator is smaller than the prey, but it can grab the prey. So remember, those are single cells; they got no brains. So it's like the didymium is using its mouth to suck paramecium into its body, its cell. And so it's a few minutes video. I'm gonna forward a little bit, and eventually didymium gonna swallow paramecium as a whole, and to finish the predation process. I think that's my last slides. I think that's it. I'm gonna stop sharing.
Okay, thank you so much for the very interesting and informative presentation.
Yeah, I think it's about 15 minutes, right? Right on mark.
Yeah. Thank you.
No problem. And how are you in New
I'm
Jersey?
doing well.
Doing well?